Hey students, just wanted to create this recording of the lesson I did this morning. This is Unit 5, Lesson 1, and in this lesson there were 4 main objectives. We got to the first 3, and so in this video I'll go over what we did this morning. So the first thing I did was I did just a quick overview of the main concepts from Unit 4, and then I spent some time giving you an introduction to the main concepts that we'll be talking about in Unit 5, as well as some vocab to get us started. And then third, I described meiosis and how it contributes to genetic diversity. We didn't get to this 4th objective. We will do this next week. So let's start with that first objective. So Unit 4, again, was all about 3 main things. It was about cell signaling and kind of cell signaling pathways. The second thing was about the cell cycle and how cell replication through the process of mitosis is regulated in the cell cycle. And then third was talking about feedback mechanisms and how they either maintain homeostasis or amplify a particular condition. Okay, so starting with that first topic, so cell signaling, one of the things we talked about in Unit 4, where there were 4 major categories of signaling, right? We have autocrine, which is where a cell sends a signal that's received by itself. We have signaling that occurs through contact or through things like gap junctions or plasmodesmata in plants. We call that juxtacrine signaling. Then we have, over short distances, paracrine signaling. And a specific example of paracrine signaling would be synaptic signaling between neurons. And then lastly, we've got that long distance signaling called endocrine, or typically this is how hormones signal other areas of the body. Okay, so with those 4 categories in mind, we actually took a closer look at how a cell signaling pathway typically works. A cell signaling pathway typically has 3 phases. It's got a reception phase, where a signaling molecule, which we call a ligand, connects to a receptor. It's typically a protein found on the membrane of a cell. Then you have the transduction phase, which is where that activated receptor kicks off a chain of events, typically a series of relay molecules or proteins like the phosphorylation cascade, to basically transmit and in a lot of cases amplify that signal to bring about the 3rd phase, which is some response. That's the response phase. And we talked about how there are many different examples of responses that can be brought about by a cell signaling pathway, whether it's apoptosis or gene activation or enzyme activation, right? All of these are potential responses of a cell signaling pathway. And like I mentioned, right, we took a closer look at this transduction phase. This is an example of a transduction phase that has a phosphorylation cascade that involves proteins called kinases, which are mainly known for phosphorylating other proteins, typically other kinases, as a means of activating those other proteins. And so like we see here, we get one kinase activating another. And there's also ways in which those kinases that have been activated can be turned off or deactivated through something called a phosphatase that we mentioned as well. While we talked about cell signaling, we talked about how there are kind of 3 major categories that we'd be familiar with, at least for this class, of cell signaling pathways. The first of which being the GPCR pathways, G protein coupled receptors, right? Typically, the example that's brought up is with the epinephrine pathway. The second major category were receptor tyrosine kinases. That was the type of receptor that has 2 monomers that when they're activated, that receptor becomes a dimer, and then that leads to multiple relay pathways being activated inside the cell. And then the third is we talked about ion channel receptor pathways, just where the receptor protein is not only something that connects to a ligand, but it also is a channel protein that opens in response to binding that ligand allowing some substance into the cell, which then leads to some particular response. So 3 major categories of cell signaling pathways, the main difference being, or at least one of the main differences being in the type of receptor. Okay, the second category of topics that we discussed in Unit 4 had to do with the cell cycle, which is represented here in this model. Okay, this cell cycle, again, is a model for thinking about how cell division is thoroughly regulated in an organism, because we want to make sure that only cells that are without damage and without errors in the DNA and that have enough resources are actually able to divide. And in this way, this prevents things like uncontrolled cell division that we see in tumor growth, different types of cancer. And this this cell cycle has 2 major phases. We have the phase in which the cell divides, which is represented by this little yellow portion. That's the M phase, or the mitotic phase, where this process that we see illustrated right here, right, prophase, metaphase, anaphase, and telophase, where that occurs. Now, the rest of this cell cycle, all the way around here, this is referred to as interphase, and this is the main component of a cell's quote-unquote life. It spends most of its time in interphase and only a small portion actually dividing. Now, within this interphase portion of the cell cycle, you have kind of 3 sub-phases. You've got the G1, the S, and the G2, and there are different events that happen within each of those sub-phases. One notable example would be in the S phase. That's where DNA replicates. But then what's more is actually, if you look within these phases, you'll see these checkpoints, and these are really where cell division is regulated. And so the 3 major checkpoints, 2 of which occur within interphase, are the G1 checkpoint and the G2, and then you have the M checkpoint, which is the only checkpoint within the mitotic phase. Now, there are different conditions that must be true of this cell in order to pass by or pass through that checkpoint and continue on in the cell cycle. So, for example, the G2 checkpoint, we learned that the cell has to have sufficient levels of MPF, mitotic promoting factor, in order to pass through the G2 and into the M phase. Okay. The last component of Unit 4 had to do with positive and negative feedback. And again, if there's one thing you can remember, is that negative feedback mechanisms serve to maintain homeostasis. And positive feedback mechanisms serve to amplify a particular condition or response. And there are multiple examples that we have for both of these types of feedback mechanisms. For negative feedback, we have something such as the feedback control of blood glucose levels, right? There are mechanisms by which a human body is able to recognize if the blood glucose levels are too low and bring about a response that raises that blood glucose back to its stable level, or if the blood glucose is too high, the body also has a negative feedback mechanism that helps to lower the blood glucose back to that stable level. The other example of negative feedback is thermoregulation. Very similar, except for body temperature. For positive feedback, there's 2 major examples that we've seen now. The first is childbirth, in particular, the amplification of contractions in response to the pushing on the cervix by the baby's head. And then we've got blood clotting in response to a wound. Typically, you see a positive feedback mechanism where activated platelets activate more platelets until you get this rapid increase in activated platelets that form a clot. Okay, So with those kind of three main parts of Unit 4, we're going to be transitioning into Unit 5, where we're going to be continuing, especially with regards to the cell cycle, to make connections in this new unit. Okay, so now that we've done our review of Unit 4, let's talk about what we're going to be seeing in Unit 5. And on this slide, there are some images that represent some of the highlights from Unit 5. Now, Unit 5 is all about heredity. In other words, how things are passed on from generation to generation. And some topics that we're going to be learning about are they're going to begin with this structure called a chromosome, which I'll elaborate on in the next section here. But chromosomes are extremely important when it comes to studying heredity, because these are where our genes are located. And what we're going to learn about is the main process by which chromosomes are passed from generation to generation, which is in the formation of these gametes, or sperm and egg, through meiosis, which is summarized right here. So we're going to learn about not only chromosomes, but also how meiosis facilitates the passing on of chromosomes through the formation of gametes. We're also going to learn about other areas of genetics or heredity by looking at one of the main experiments that was done to begin our understanding of how traits are passed down from generation to generation, and that was with Mendel's pea plant experiments, which you might have heard of in your regular biology class. And so we're going to learn about things like dominant and recessive traits. We're going to learn about how we can predict the traits of the offspring between two, in this case, pea plants, or just two organisms, if we know what forms or form of the trait that they have. We're also going to learn about a tool called a pedigree that's going to help us to make predictions as to whether or not a trait is dominant or recessive, and actually which chromosome it's most likely located on. And then the last thing that we're going to use a lot in Unit 5 is we're actually going to use a lot of statistics to analyze the results of different genetic crosses to actually make predictions that help explain results that maybe we don't expect for particular genetic crosses. And so we're going to learn about something called the chi-square test, which is a helpful tool for determining if the results of a genetic cross are in line with what we would expect following typical core principles of genetics that are based off of Mendelian genetics or the pedigree analysis. And so we're going to use the chi-square tool, which you learned about in your summer assignment, to actually be able to make those predictions. Okay, so these are just a few of the main concepts that we're going to see in Unit 5, things like chromosomes or meiosis or pedigree analysis, or looking at genetic crosses and statistical analysis to understand those crosses will be the highlights. So now that we have a little bit of an idea of what we're going to talk about in Unit 5, let's talk about one of the first main topics, which is meiosis. Okay, and in order to talk about meiosis, we got to talk about a few key vocab terms, and these are in your notes as well. So I mentioned, right, one of the main things that we're going to be referring to a lot in this unit is a chromosome. And a chromosome, if you remember, is a tightly wound strand of DNA that forms these structures that we see here and also down here. Now, what I really liked about this figure right here is it shows that this DNA is actually wrapped very tightly around this protein that's called a histone. And that's what a histone is. It's these little purple spheres that are They help facilitate the tight wrapping, the tight coiling of DNA that we see in a chromosome. Now, how is a chromosome related to a gene? Well, a gene is just a section of the DNA. And so a gene, like we see right here, is a section of that DNA typically that controls a particular trait. And another term that we use to kind of describe the location of a particular gene is the term locus. And so we would say that the locus of this particular gene is right about here on the chromosome. That's just the specific location of a section of the DNA on a chromosome. Okay. Now, you've likely heard before, right, that humans, we have 23 pairs of chromosomes, and we can actually organize these into a useful diagram called a karyotype. A karyotype, which is represented over here. We see 23 pairs of chromosomes. 1 through 22 are numbered. The 23rd pair we refer to as our sex chromosomes. This is showing the sex chromosomes of a biological male, biological female being XX. And we also distinguish between sex chromosomes and the other 22 pairs. We call the first 22 pairs of chromosomes, we call those autosomes. So that's just a non-sex chromosome, whereas the X and the Y chromosomes are sex chromosomes. Okay. Now, some other terms that we're going to use that are going to be useful when we talk about meiosis are homologous chromosomes and sister chromatids. So I want to talk real briefly about the difference between those. So first, let's start with homologous chromosomes. Okay, so a homologous pair of chromosomes, or homologous chromosomes, refers to, if we look at this chromosome number 1 in this karyotype, we know that we get half of our genetic information from our mom and half from our dad. And what that means is that, remember, we have 23 pairs of chromosomes. For each chromosome number, that means we get one of that chromosome from one parent and the other copy of that chromosome from the other parent. And so we refer to this as a homologous pair, meaning that it is representing the same chromosome number, although as we'll see in this unit, that there can be slight differences between homologous pairs of chromosomes. Now, the difference is when we say sister chromatids, right? So sister chromatids and homologous chromosomes are different. The basis of a sister chromatid is when our DNA has been duplicated. And let me show you what I mean. So whether it's in meiosis or mitosis, before that process occurs, our DNA has to be duplicated, particularly in the S phase of interphase. I've added here, this is another karyotype, but you'll notice if you compare these, right, look at chromosome 1, right, what's different is that the homologous pair has been duplicated. And so we say that the identical copies of one part of the homologous pair, those are two sister chromatids. So if I were to label that, so these two right here, right, those are identical, right? And so would these two. And so we say that these are sister chromatids, and these are sister chromatids. Okay. Now, that just means that they are identical. And you see that represented down here as well, right? These chromosomes, I believe there are 3 chromosomes represented down here. They've already been duplicated, and you'll see sister chromatids and homologous chromosomes each labeled in that diagram. Okay, so now that we've got those terms down, let's start to talk a little bit about meiosis. Okay, so on this slide here, on the left-hand side, this is a really good summary of the process of meiosis. So what we need to know is that meiosis, its major function is to produce gametes, which again are reproductive cells like sperm and egg cells. Now, looking at this diagram, right, we start with, at the beginning, a pair of homologous chromosomes. Now, in reality, remember that we've got 23 pairs of chromosomes, but just for the sake of this diagram, they only show one pair of the chromosomes. And what we have happening in the interphase, right, is that the chromosomes are duplicated during the S phase. And so here we have our sister chromatids, or in other words, we've got the identical copies of each part of this homologous pair. And there are two halves to meiosis. There's meiosis 1 and meiosis 2. The key thing to remember about meiosis 1 is that we have a splitting of the homologous chromosomes. A splitting of the homologous chromosomes, but in meiosis 2, we split the sister chromatids. So if there's one thing to remember about meiosis 1, we separate homologous chromosomes. In meiosis 2, we separate sister chromatids. And what we end up with, we refer to these as haploid cells. And I like to think of the term haploid as referring to half of the original genetic information, or in other words, one set of the chromosomes. And the other term that you'll see that looks similar is diploid, and that just refers to two sets of the chromosomes. Right, so if you remember, right, a human has 23 pairs. So I like to think of, in terms of the pairs, if you just have one chromosome from each pair, in other words, the contribution from one parent, that is one set of the chromosomes. But since we have 23 pairs, we actually have two sets of the chromosomes, 23 pairs. And so when we have two sets, we call that diploid. But at the end, right, at the end of meiosis, we have haploid cells that have only one set, because what we're trying to do is create gametes that will combine a sperm with an egg that each contribute one set of the chromosomes to that offspring to then have two sets to become diploid again. Now, if you look on these other figures, you're going to see just a little bit more detail about these two processes. So meiosis 1, meiosis 2. What I want you to focus on here is actually within metaphase of both of these parts of meiosis. This is kind of the major difference between these two processes, right? You'll see in metaphase 1, right, they line up in the center, very similar to in mitosis, right? We have the chromosomes lining up in the middle, but you'll notice, right, they are lining up in homologous pairs. And when it splits, or when the chromosomes begin to be pulled away in anaphase, right, you'll notice that those homologous pairs are being separated, but you still have those sister chromatids are together. Now in metaphase 2, that's where those chromosomes line up again, and then the sister chromatids separate, leaving you with that haploid cell at the very end, 4 haploid cells at the end. Okay, so that's an overview of the process of meiosis, where we also use those terms homologous chromosomes and sister chromatids. Now, one thing just real quick to note, and I'll talk about this more next week, I mentioned that meiosis produces gametes, which are sperm and egg cells. Mitosis is typically used for replicating somatic cells, which I just like to think of non-gamete cells. So like a skin cell would be a somatic cell. That's what I mean by that term. Okay. The last thing that we'll talk about here is how meiosis contributes to genetic diversity, right? Meiosis is this core process that's key for sexual reproduction. And there are two main ways that meiosis actually increases genetic diversity based on two major events that happen. So the first is something called independent assortment. And the idea here, if you look at this figure, is that during metaphase 1, remember, which is where the homologous pairs of chromosomes line up down the middle, there are actually multiple orientations for how those chromosomes will align, like we see here, right? And that will ultimately affect which chromosome the gamete at the end receives. Now, if you remember, we've got 23 pairs of chromosomes, so that means for every pair of chromosomes, right, there are multiple orientations for how it can align on that middle line during metaphase 1. Now, the other way that meiosis increases genetic diversity is through something called crossing over. And we'll talk about more around this topic later this unit, but the idea here is that actually during prophase 1 and metaphase 1, again, where those homologous duplicated chromosomes are lined up, an event called crossing over can actually lead these homologous pairs of chromosomes to swap portions between non-sister chromatids, leading to what we see right here, right, these recombinant chromosomes, which ultimately increase the variation of the gametes. So independent assortment and crossing over are two parts of meiosis that both increase the variation of the gametes and ultimately the variation of a population. Okay, so we will talk more about this next week, but hopefully if you missed any part of the lesson today, you were able to catch up by rewatching this. Have a great weekend.